In the last video, we discussed how glomerular filtrate is produced at the glomerulus and moved into the nephron tubule there at the glomerular capsule. In this video, we will look at the mechanisms that convert the very dilute glomerular filtrate into much more concentrated urine so that we are conserving most of the water that squeezes out of the glomerulus and into the filtrate. Excuse me. So let us begin the process. Okay, so the very first, or the first of the two processes that we're going to discuss is tubular reabsorption. And again, to remind you, tubular reabsorption is movement of materials from the nephron tubule back into the interstitial fluid and then into the blood. So we reabsorb materials that are found in glomerular filtrate and bring them back into the body through the process. And there are three ways that materials move when we do tubular reabsorption. The first is diffusion, and hopefully everybody now remembers what diffusion is. We move materials by diffusion from what concentration to what concentration. Give you a chance to think about it. Hopefully you said from high concentration to low concentration. And they move. Is ATP required? Think about that one. With diffusion, no ATP is required. So lipid-soluble materials just move from regions of high concentration to low concentration because of the concentration gradient, not because we're using any kind of ATP-driven process. The second process is facilitated diffusion or facilitated transport. That is when diffusion occurs, again from high to low concentration, but this time no ATP is still required, but this time there is a carrier molecule that is moving the material, so it's not just being transported passively across the cell membrane because of the concentration gradient. It's being transported across the membrane because there's a carrier molecule there that will bring it from one side to the other. And then the third process is active transport. And this allows us, because we're using ATP, this allows us to move materials against the concentration gradient from a low concentration to a higher concentration. So those three processes, diffusion, facilitated diffusion or facilitated transport, and active transport, are all being employed to shape the tubular fluid through reabsorption. So once materials are reabsorbed into interstitial fluid, then they move on into the peritubular capillaries and back into the circulatory system. So and the ability of molecules to diffuse is enhanced because, first of all, there's very low blood pressure in those peritubular capillaries. Now remember, we've taken away most of the fluids, and with a lot of the fluids gone, blood pressure is going to be greatly reduced in the peritubular capillaries. And so that, because there's very little pressure pushing fluids out of the capillaries, most of the movement is going to be back into the capillaries. So low blood pressure prevents, it assists fluids and materials to move into those capillaries that are surrounding the, everything, that all the capillaries that are beyond the efferent arterial are going to have low blood pressure until fluids are restored. And then because of the colloid osmotic pressure of the blood in those capillaries. That's going to draw fluids into the capillaries. And then the last characteristic of those peritubular capillaries is that they are really permeable. The pores between the cells are very large in the peritubular capillaries, and so that allows fluids to move much more quickly as well. So 3 characteristics of the peritubular capillaries really assist in moving materials into them. So where do we do tubular reabsorption? The bulk of tubular reabsorption occurs at the proximal convoluted tubule. And this makes sense. This is where the fluid is going to be the most dilute. So this is where a lot of the water is called back into the body through tubular reabsorption. It's further enhanced because the proximal convoluted tubule cells, those cells have microvilli, referred to sometimes as the brush border, because when you look at it underneath a microscope, it has this kind of the microvilli appear to be like little hairs, and so that brush border greatly increases the surface area of the cells that line the proximal convoluted tubule. Just like, in exactly the same way that in the digestive system, the microvilli also increase the surface area. And so here at the proximal convoluted tubule, we reabsorb any glucose, amino acids, or any small proteins that have escaped the blood back at the glomerulus. So again, the goal of the glomerular capillaries is that they maintain proteins, but sometimes a few escape. And so we have capacity at the proximal convoluted tubule to reabsorb glucose, amino acids, and proteins. As well, creatine, remember a byproduct of muscle metabolism, creatine, and metabolic acids. These are acids that are produced because of body processes like citric acid, uric acid, lactic acid, and ascorbic acid. These 4 acids are all drawn back into the body by tubular reabsorption. We only see these materials, glucose, amino acids, the metabolic acids, in urine if they are present in filtrate at such great amounts in such large concentrations that they overwhelm the ability of the carrier molecules that are found in the proximal convoluted tubule to bring them back. If we're pushing so much of these materials through the proximal convoluted tubule that the carrier molecules that are found there don't have time to reabsorb all of it, that some of it escapes. So that amount, the amount that the proximal convoluted tubule can reabsorb, is referred to as the renal plasma threshold for that carrier, for that molecule. So, for instance, people who have diabetes, if it is uncontrolled diabetes, where blood sugar gets so high that there isn't time, that the glomerular filtrate does not remain in the proximal convoluted tubule long enough to gather back all the glucose. So lots and lots of glucose gets reabsorbed by people with diabetes, but there is just so much glucose in a person with uncontrolled diabetes. There's so much glucose in their blood that that the proximal convoluted tubule just can't reabsorb it all. And so once that glucose passes beyond the proximal convoluted tubule, there are no carrier molecules that can be involved in that, that can do that reabsorption in the rest of the renal tubule, and so we've exceeded the renal plasma threshold for glucose in a person who has uncontrolled diabetes. Okay, so most of tubular reabsorption occurs at the proximal convoluted tubule. Sodium ions, and remember, sodium ions have a positive charge. So as sodium pumps bring sodium into the proximal convoluted tubules, or reabsorbs, I'm going to try that again, apologize. As sodium ions are reabsorbed by the carrier molecules, the sodium pumps in the proximal convoluted tubules, lots of negatively charged electrolytes like chlorine are also brought along with it. That creates such a such a voltage difference that it just draws the chloride ions across the membrane of those cells. And as we move sodium and other electrolytes across the renal tubule, that creates an osmotic gradient that also draws water across the proximal convoluted tubule wall, so that by the time all of the fluid, the filtrate, is leaving the proximal convoluted tubule, the tubular fluid, the developing urine, is isotonic. It has the same osmotic strength as interstitial fluid. So the upshot of tubular reabsorption is that by the time the urine is built at the far side of the nephron tubule, almost all of the sodium and almost all of the water, remember 99%, and I might even add that greater than 99% of the water that we squeeze out of the blood at the glomerulus has been reabsorbed into the body. Okay, so again, the bulk of reabsorption occurs at the proximal convoluted tubule, but we do continue to reabsorb materials throughout the whole entire renal tubule. So there is a table, and I don't have my book with me. I'm in Texas still, and I don't have my book with me, but there's a table in the book that describes all of the places that tubular reabsorption occurs and what materials are reabsorbed. And so I want to encourage you to find that table in the chapter, because that table is going to be very useful to you. And I'm going to try to also send out an email or an announcement with that information in it. So look for that as well. We also reabsorb water at the distal convoluted tubules and at the collecting ducts. So there's two more locations where water reabsorption occurs, at the distal convoluted tubule and also at the collecting ducts. Okay, so some terms that relate to tubular reabsorption. Diuresis means an increase in the volume of urine. So a diuretic is a material that causes increased urine volume. Diuresis, greater volume. And if we increase glucose or protein in tubular fluid, so if for some reason we cannot reabsorb all the glucose or proteins that are found in tubular fluid, or if there is damage to the glomerulus so that tubular fluid contains a bunch of protein, which is also a consequence of diabetes, then all of the glucose and the proteins or proteins in the tubular fluid create osmotic pressure that draws fluids in back, that prevents the reabsorption of water, causing osmotic diuresis, or increased fluid content because of the osmotic pressure of the tubular fluid. So we wind up retaining or maintaining, that's probably a better choice. We wind up maintaining more water in the urine because there's more dissolved materials in the urine. Okay, so the second process then that converts tubular fluid into urine is tubular secretion. And recall, tubular secretion is movement of materials from interstitial fluid into the developing urine so that we can further excrete materials that we no longer want in the body or that are waste materials to the body. So we are capable of secreting specific organic compounds. So remember, an organic compound is anything that contains carbon and hydrogen at minimum, but also usually oxygen. So these include pharmaceuticals. So penicillin and phenobarbital are both materials that are secreted into developing urine, and metabolic wastes like creatinine, ammonia, and histamine. These are materials that we don't need in high concentrations. So hydrogen ions, for instance, are actively secreted into the developing urine, and it requires active transport to do this. So ATP is required to secrete hydrogen ions. And potassium ions are also secreted. So we can secrete organic materials that are waste products to the body. We can secrete hydrogen ions to maintain acid-base balance. And things like potassium ions are